Assalamu alaikum wa rahmatullah. Today I will start with you a new topic, which is signal transduction. It is going to be covered in about 5 lectures, and today I will introduce you to the topic, and we will continue after about 2 weeks. بنكمل بعد أسبوعين ونص تقريبا برجع لكم نكمل signal transduction. So today it's going to be only the first lecture of signal transduction.
So, in this set of lectures, we will define different mechanisms for signal transduction, and this is actually to understand the way hormones can induce their effects in our bodies. So the scope of these lectures is to understand the mechanisms for hormones actions. We will identify different hormones types in terms of chemical structure or in terms of their characteristics and mechanisms of action and the type of signaling that they use. We will describe many receptor types and examples from our hormones. We will outline the hormone receptors interactions. Also, we will identify different types of second messengers that are used by different hormones in order to induce their effects in our body. So we will identify many examples for different signal transduction mechanisms in our body. So at first we will define some of the terms that we will be learning in these lectures. Also, we will identify the different components that are involved in signaling, and we will identify different types of signaling. Types of signaling ligands. Also, we will identify the types of different ligands, especially their chemical characteristics. We will identify different types of signaling receptors. There are many types of signaling receptors, such as ion channel linked receptors, G protein coupled receptors, enzyme linked receptors, such as tyrosine kinase receptors, and we will identify their main mechanism. Also, we will look at different second messengers. We will identify what does second messenger mean and what are different types of second messengers. And of course, we will learn some of the examples of different signaling mechanisms in our body that our hormones use to induce their effects in our body. So, first of all, what do we mean by signaling? Signaling between cells. It means cell-to-cell communication via signals. إذن هي التواصل بين الخلايا عن طريق إشارات. Signal transduction is a process of converting an extracellular signal into intracellular response. لما نحكي signal transduction من كلمة transduction نعرف إنه في صار عملية conversion للسيجنال، توصيل للسيجنال from outside the cell to inside the cell, which is represented by a certain response or a change in the activity of the cell. Okay, so what does ligand mean? Ligand is a signaling molecule. إذن هو الجزيء اللي بده ينقل أو الإشارات هي الإشارات اللي رح تنقل المسج من خلية إلى أخرى. هذا هو اللايجند. لما نحكي ligand إذن في receptor. The ligand binds to a receptor. A receptor is specific protein that binds a specific ligand in order to transmit or conduct this signal to intracellular target. إذن الريسبتور عبارة عن نوع من أنواع البروتينز that has a binding site for the ligand. Okay. هلا الوظيفة تاعت الريسبتور هو to transduce أو transmit the signal الإشارة من الخارج إلى الداخل. And there are so many different receptors that can respond differently to the same ligand. فالريسبتور هو اللي بيحدد الأكشن. أوكي، يعني حتى لو اللايجند نفسه ارتبط بأكثر من نوع من الريسبتورز we will find different actions. ليه؟ لأنه الأكشن depends not only on the ligand but also on the receptor. Now, the main components that are involved in signaling ligands, the receptors, كمان في different intracellular signaling proteins. then في ست كاملة من البروتينز اللي involved في السيجنالينج. تبدأ من اللايجند اللي هو نفس السيجنال والريسبتور اللي بيرتبط فيه اللايجند. بعدين في other intracellular signaling proteins involved. فزي ما انتم شايفين هون هدول الكومبوننتس of the signaling. في عنا السيجنال اللي هو ممكن يكون الهرمون وهذا الريسبتور. اوكي، بعدين في different signaling proteins اللي هم mediators بنقولوا السيجنال. اوكي، ممكن يكون من بينهم اشي بنسميه second messenger. at the end هنا اللي هم target proteins اللي هم رح يصير فيهم change. طبعا التارجت تاع السيجنالينج ممكن يكون activity of an enzyme or it could be gene regulatory protein in order to change the expression of a certain gene, or it could be a cytoskeletal protein which will change the structure of the protein or maybe movement or the shape of the cell. So these are different targets of the cell signaling. So we have intermediary proteins, intracellular signaling proteins, we have enzymes which induce different metabolic activities in the cell. We will have also second messengers. We will identify second messengers in the coming lectures. We also have target proteins. We have inactivating proteins. So we will come across all of these terms in the next lectures when we start talking about each type of signaling mechanism. So signaling, what is the main function of signaling? Why is signaling important? Why are we studying signaling in this introductory course? Signaling actually define how cells can respond to their environment and how can they differentiate or change over time. So signaling is very important for the cells to maintain living because cells are found in a changing environment. The environment surrounding cells is not constant. There is dynamicity in the environment, سواء كانت the internal environment in our bodies or the external environment surrounding our bodies. So the environment is always changing. So the cells must respond to these changes in order to maintain homeostasis, and I'm sure you are familiar with the term homeostasis. So cells always try to adjust to their environment, and that happens due to signaling between the stimulus and between the cell. So this is the way cells can detect and respond to their environment by signaling. So this is the first, let's say, the first function of signaling, which is to respond to the environmental changes and respond to these changes. For example, changes in oxygen availability, in sugar availability, or in temperature. Second, sometimes the signal it is not just only a switch on and off. Sometimes we will have graded signals, and these signals may induce cells to differentiate into different types of cells. So signals is not an on-off switch only to the cells. There are so many different types of signaling that can take place. Some of these signals, their intensity can have different intensities of certain signal can have different outcomes or different effects on cells. And that's what we may see in the differentiation of cells. تمايز الخلايا ممكن شدة السيجنال تأثر على نوع الخلية اللي راح تتميز لإلها. The third example here, sometimes we have combined actions of different signals and factors can create different cell types, and that also we see in differentiation. So we are not talking about one signal per cell. We're talking about a combination of signals that have different actions and different effects on transcription factors. So different combination can create different types of cells, and that's also important for cells differentiation. The fourth example for signaling is that sometimes the signal can be inhibitory. Signals are not always excitatory. So sometimes the signal can be inhibitory for a certain cell to maybe stop duplication or stop proliferation of certain type, for example. And finally, different signals, they integrate and then together can help the cell to adjust to some external changes to the external environment, or to act differently or change with time. So signaling is very important for how the cells can respond to their environment and how can they change over time or differentiate. So what is the main mechanism for signaling? The main scenario for cell signaling is that we have a ligand here that was secreted by maybe a hormonal secreting cell, hormone secreting cell, ممكن تكون gland. This ligand will find a receptor that is specific for this ligand on the cell, maybe on a neighboring cell, and then will bind specifically to that receptor. Now, this receptor binding will induce changes in the receptor, maybe a conformational change in the structure of the receptor, which will induce other proteins that can bind to the receptor inside the cell. So now the binding of the ligand transduced changes at the intracellular level, and these changes will be transduced into different target proteins in the cell. So these mediators we can call second messengers sometimes. Okay, so these second messenger will transduce these changes into a cellular response. Okay, maybe increasing activity of a certain enzyme that will produce a certain or activate a certain product or produce a certain substance to the cell. Or it can, this second messenger can go directly into the nucleus to induce changes in the gene expression and in production of maybe receptors or enzymes or different proteins, maybe structural proteins. So as you can see here, signals that are outside the cell, they got translated into cellular response. Okay, and without even having to transport this signaling molecule into the cell. That's why we call this transduction. Okay, so we don't necessarily need to transport the molecule itself into the cell to induce its actions. So only binding to the receptor can induce these actions. So the question now is, do we need all receptors to be bound to their ligands in order to have a certain physiological response? So that is an important question. We will answer that looking at this graph. Okay, on the x-axis we have the relative concentration of a ligand. So we have like 1x normal, 2x normal, 3x normal, 4x normal, the concentration of the ligand. And on the y-axis we will see the fraction of maximal binding of the or the cellular response. So we have two curves. The one in blue, okay, it demonstrates the physiological response. It could be like contraction of a muscle. And the purple line demonstrates fraction of surface receptors that are bound with their ligand. Okay? So let's see how the fraction of surface receptors bound to the ligand relate to the physiological response. So let's take the first concentration, which is the normal concentration of a ligand. You can see that even with 50% of receptors bound to their ligand, we can have nearly maximal, close to maximal physiological response. Okay, 0.8. or 80% of the physiological response. However, we only have 50% of the receptors occupied or bound to their receptors. Look at the double, okay, concentration of the ligand. Of course, will induce more binding. So we will get about 70%, let's say, or 65% of the receptors bound to their ligand. The physiological response is maybe 90%. Okay, so even at lower concentration of the ligand, we could achieve very close to the maximal physiological response. And even when we doubled the amount of the ligand, we didn't get that much increase in the maximal response. Okay, it only increased for maybe less than 10%. So what the take-home message here is, we don't need to bind or to have all the receptors to be bound to their ligands in order to induce a good response or a good physiological response. So there is a reason for that we come across. Another characteristic of signals, that signals can act locally or at a long distance from the secreting cell. So in the first hormone on the right, we will have this example of signals secreted by neighboring cells, and the target is a neighboring target cells. So the signal acted locally, acted through a short distance from the secreting cells, and then bound to receptors on the target cells that are very close to the secreting cells. And in this case, we call these signals local mediators. Okay, local mediators or local factors. Another example of another signal, sometimes signals are secreted from the secreting cell or a gland and then are transported via blood or through the circulation into a distant target cell. إذن ممكن إنه السيجنال تنتقل إلى الخلية التارجت تكون بعيدة جدا. فمثلا ممكن تكون عبارة عن هرمون secreted من البيتويتري جلاند اوكي بالبرين، وهاي السيجنال to be transported or carried by the blood and then can go to a distant target. اوكي هذا التارجت ممكن يكون مثلا في خلية مثلا عضلية أو عظمية موجودة في القدم for example. So signals can act either locally or at distance. طبعا الاندوكرين هرمونز بيشتغلوا بهاي الطريقة اللي هي طريقة النقل عبر البلد. endocrine hormones بينتقلوا عن طريق البلد. there is no duct، عشان هيك سميناهم الغدد الصماء، ما في duct إلهم حتى ينقلوا السكريشنز تاعتهم، لكن البلد هي الوسيلة تاعت النقل إلهم. فهذا example على الإندوكرين هرمونز. Okay. What about the responses? The response of a signal, is it always fast or always slow? It can be fast or slow, okay, depending on the response itself. So let's see this schematic. We here have a classical signaling molecule that binds to a cell surface receptor. Look at the downstream signaling اللي صارت هون. بالريد لاين صار في altered protein function، يعني البروتين function تغير. مثلا صار في activation لإنزيم معين، هذا الإنزيم رح يعمل function معينة بالسيتوبلازم. اوكي، مثلا رح يغير direction او الشيب تاع السيتوسكيلتون تاع اللي جوا الخلية. اوكي for example، اما مثلا رح يزيد البايندينج of certain cytoskeletal proteins together، فبالتالي رح تتغير السيتوبلازميك ماشينري، وبالتالي رح يصير في تغير بالسيل behavior. So this is fast. It can be the duration can be from seconds to minutes لأنه already البروتينز موجودة لكن غيرنا الانتراكشن بينها. هلا السكند سيناريو اللي هو بالجرين لاين رح تلاحظوا إنه السيجنال is transduced into the nucleus where gene expression now is modified. ممكن يكون صار في activation لا expression of a certain part of the gene that will be translated into RNA and then into generation of new proteins or peptides that will change the let's say the cytoplasmic machinery أي organelle موجود في الcytoplasm which will induce a change in the cell behavior or function. That actually is slow because it takes minutes, hours, or days in order to get these changes. So that's why our answer to this question is that the response to the signal can be fast or slow depending on the route or on the mechanism that was induced to make these changes. دايما لما تكون gene expression it requires hours or days. But if we only need to change the activity of an enzyme or a protein that is there already, we just change the activity of this protein. This actually is very fast. For example, if we only phosphorylate an enzyme in order to induce its activity, this is very fast. Can happen between seconds to minutes. Now, a very important characteristic of signals is that the signal can be amplified. For example, here we have epinephrine that was, let's say it was secreted from the adrenal medulla due to sympathetic nervous stimulation. For example, في حالة stress أو توتر فطلع epinephrine from the adrenal gland. The concentration of epinephrine was 10 to the minus 10 molar. Okay, 10 to the minus molar أقل من nanomolar الconcentration تاعه. Now, this epinephrine will bind to receptors that will activate an enzyme called adenylyl cyclase موجود على السيل membrane. هذا example. طبعا احنا رح نعرف كل أنواع pathways إن شاء الله coming lectures. But this is only an example to understand this characteristic of signals. So لاحظوا إنه السنجل أو هذا الكونسنتريشن من الإبنفرين activated many adenylyl cyclases. Okay, each one of these adenylyl cyclases will generate cyclic AMP. Okay, and then we will get a concentration of cyclic AMP that is 10 to the minus 6. Okay, so we started with epinephrine with a concentration of 10 to the minus 10, and then we got a second messenger called cyclic AMP. And cyclic AMP, which is a signaling molecule, is with a concentration of 10 to the minus 6. ليش؟ لأنه عندنا many adenylyl cyclases generated more and more cyclic AMP signals, and the same adenylyl cyclase will produce more than one molecule of cyclic AMP. Each cyclic AMP will bind to an enzyme called protein kinase A, and protein kinase A will be able to phosphorylate and activate many enzymes. So each cyclic AMP will activate a protein kinase, and each protein kinase will activate many enzymes. So by the end of this signaling cascade, we end up having the signal amplified. اتضاعفت السيجنال هاي، اتضخمت. Okay, فإحنا we started with very little and we ended up with a very high and aggravated signal. Okay, so now we can answer the question of, do we need all the receptors to be bound to their signals in order to have a good, a fair physiological response? No. Why? Because signals are amplified. Okay, so even if we started with very low concentration of a ligand, we will end up having so many molecules that has been generated as second messengers, and so many enzymes that have been activated to induce the response that we need. إذا هذا بيجاوب لنا على السؤال إنه ليش إحنا ما بنحتاج إلى إنه إحنا نعمل إشباع تام لكل الريسبتورز لحتى إنه نحصل على physiological response مهمة، مثلا contraction of a muscle. Now let's talk about the different types of signaling. The first type of signaling is the contact dependent signaling. So this signaling takes place when we have two neighboring cells that are in direct contact. Their plasma proteins, they contact directly as a ligand to receptor binding. Okay, so both of these cells are in direct contact via binding of plasma membrane proteins. So this is what we call contact dependent signaling. The second type of signaling is the paracrine signaling. When we have a cell that is secreting factors or signals, these factors or signals travel a very short distance, okay, يعني مسافة صغيرة جدا into the target cells. بتلاقي الريسبتورز السبيسيفيك لهذا اللايجند على التارجت سيلز اللي هم in the neighborhood, then they bind and induce signaling in these target cells. So الباراكرين signaling بيكون بخلايا قريبة من بعض لكن they travel very short distance. هذا اللي بنسميه الباراكرين أو مرات بنسميه local mediators أو local factors. هلا الثيرد تايب اوف سيجنالينج اللي هو الساينابتيك، اوكي، هو بيشبه الباراكرين لكن اللي بيفرقه انه هو بين نيورون، اوكي، يعني في ساينابس. لما نحكي عن ساينابس بده يكون في نيورون عمل ساينابس مع نيورون او مع تارجت سيل، اوكي، ممكن تكون سكيليتال ماسل او مثلا كاردياك ماسل. فهلا الاكسون بيطلع نيورو ترانسميترز في الاكسونال تيرمينال. هاي النيورو ترانسميترز هي السيجنالينج موليكيول، رح ترتبط طبعا على سبيسيفيك ريسبتورز او channels لهم ريسبتورز، اوكي، اللي هم بنسميهم ligand gated channels على التارجت سيلز، ورح يعملوا نوع من السيجنالينج. هذا بنسميه ساينابتيك لانه في ساينابس اكشلي. هلا اخر نوع من السيجنالينج اللي هو اهم سيجنالينج اللي احنا بدنا نحكي عنه بالفيزيولوجي this introductory course اللي هو السيجنالينج اوف endocrine hormones. الاندوكرين هرمونز، هلا في عنا جلاند او endocrine cell موجودة في مكان، مثلا في البيتويتري، مثلا البيتويتري جلاند في البرين. و it will secrete molecules. هاي المولكيولز are carried by the bloodstream، إذا تحمل بالبلد، and then circulate through the body to reach a target cell that is distant from the secreting cell، يعني رح تكون كتير بعيدة ممكن عن الخلية اللي هي الجلاند، and then will find a receptor على هاي target cell and then will induce signaling in the distant target cells. فهذا بنسميه endocrine لأنه الهرمونز أو السيجنالز اضطرت إنها تمشي في البلاد ستريم، ما في دكت توصل بين الاندوكرين سيل والتارجت سيل. إذا الوسيلة هون للنقل الهرمون هو البلاد ستريم، وهذا هو تعريف الاندوكرين جلاند، إنها بتفرز السكريشنز تبعتها and transported via blood. أوكي، ضل آخر نوع من السيجنالينج اللي هو الأوتوكرين. هلا الأوتوكرين رح نرجع نشرحه برسمة بسكيم جاي بالسلايدات. الأوتوكرين لما السكريشنز أو السيجنالز تاعت الخلية ترتبط بريسبتورز binds receptors on the same secreting cells، يعني نفس السيجنالز اللي طلعت من السكريتوري سيل will bind receptors on the same cell، بتأثر على نفسها، عشان هيك بنسميها أوتوكرين. ليه؟ لأنه هاي سيجنالينج ذاتية. أوكي، غالبا بتكون هذا النوع من السيجنال نوع من الفيدباك للخلية. أوكي، فمثلا في أغلب الأحوال معظم الأحوال لما الخلية تفرز كمية كبيرة من سيجنال معين، هاي الكمية الكبيرة بترتبط بريسبتورز على السيل ممبرين to inhibit السيل. إذا مش كل الأحوال، معظم الأحوال، فبصير عندنا نوع من النيجاتيف فيدباك، إنه خلص كفاية السكريشنز. هلا نحكي عن ال types of ligands، types of signaling ligands. هلا في two main types of signaling ligands. أول type اللي هو ligands that bind to cell surface receptors. إذن في ligands بيرتبطوا ب receptors اللي هم integral proteins موجودة على السيل ممبرين، وإلهم extracellular domain في binding site بيرتبط specifically بهذا اللايجند. مثال عليهم مثلا النيوروترانسميترز اللي هم النواقل العصبية، البيبتايد هرمونز، أوكي، مثلا الجروث فاكتورز، اللايبوفيليك، مرات بعض اللايبوفيليك signaling molecules مثل البروستاجلاندينز. الميزة في معظم هاي اللايجانز إنهم إما إنهم charged polar they cannot cross the plasma membrane، أو peptides كمان large ما بيقدروا يدخلوا الممبرين. أوكي، مثل الجروث فاكتورز كمان ما بيقدروا يدخلوا الممبرين، أو ممكن يكونوا lipophilic signaling molecules لكن كمان بيرتبطوا بريسبتورز. إذا في غالب الأحوال they cannot cross the membrane، لكن مش في جميع الأحوال. أوكي، بعض الأحيان بيكونوا lipophilic molecules لكنهم charged فما بيدخلوا. أوكي، هلا السكند type of ligands اللي هم they bind to intracellular receptors، يعني they can freely cross the plasma membrane, they can diffuse by simple diffusion and then interact with intracellular receptors in the cytosol or sometimes even to the nucleus. مثال عليهم اللي هم الستيرويد اللي هم lipophilic lipid soluble chemicals، الثايروكسين كمان بيقدر إنه يدخل directly very small retinoic acid، النيتريك أوكسيد اللي هو غاز كمان very easy to diffuse across the plasma membrane. فهدولا they have the option that they can freely cross the plasma membrane and then bind directly to their receptors inside the cell or inside the nucleus. So, as we discussed previously, there are two types of hormones. Hormones that affect locally, okay, which we call paracrine or autocrine. So they affect, or their target cells are present locally, very close to the secreting cells, or في الأوتوكرين اللي هي the same secreting cell will be affected. The second type is circulating hormones اللي هم الendocrine hormones, when the signaling molecules travel via the blood circulation and then affect the distant target cell. So now let's look at the chemical classifications of hormones. Hormones are divided into either lipid soluble or lipid insoluble hormones. So for the lipid soluble hormones, they actually can easily diffuse across the plasma membrane, okay, such as the steroids. However, in the plasma, when they are transported, they have to use transporting proteins in order to be transported in the plasma, okay, because they are lipophilic. So they don't like the media of plasma, which is water mainly. So they need transporter proteins in order to be transported via the plasma. But when they reach their target, they can diffuse across the plasma membrane, such as the steroids, which are lipids derived from cholesterol. They are very or highly lipophilic hormones. Examples of steroid hormones are testosterone, estradiol, which are the 6 hormones. Also, we have the cortisol or cortisone and progesterone. All of these are lipophilic hormones or steroid hormones. Now, a second group of lipophilic hormones are the thyroid hormones, which are amine but lipid soluble. إذن هم amine chemicals لكنهم lipid soluble. The last group is the nitric oxide, which is a gas and also highly permeable. Okay. The second chemical class of hormones is the water soluble class, or in other terms, the lipid insoluble hormones. Those actually circulate freely in the plasma. They don't circulate bound to plasma proteins. They are found free from plasma proteins. Examples for these hormones are the amines, which are hormones derived from tyrosine and tryptophan. Also, polypeptides and proteins. Polypeptides are peptides that are less than 100 amino acid in length, مثل الانتيدايوريتيك هرمون. Protein hormones اللي هم larger polypeptide with more than 100 amino acid in length مثل growth hormone. Another hormones that are water soluble are the eicosanoids. Those are lipophilic, derived from arachidonic acid. They are about 20 carbon length. لكنهم غالبا بيكونوا موجودين charged at physiological pH. They are considered water soluble. They require actually, they cannot cross the plasma membrane by simple diffusion. Okay, so من ضمنهم water soluble or lipid insoluble substances, ومثال عليهم prostaglandins.
One of the lipophobic or hydrophilic hormones are the glycoproteins. Glycoproteins are actually long polypeptides. They have more than 100 amino acids, but they are bound to one or more carbohydrate groups. Examples of this group of hormones are the follicle stimulating hormone and luteinizing hormone, which are the female sex hormones. Also the TSH, thyroid stimulating hormone, as well as HCG, which is the pregnancy hormone, human chorionic gonadotropin hormone. Those are examples on glycoproteins. They have subunits, for example, alpha and beta, and this is a very important group of hormones. Now, hormones also can be divided into polar and non-polar. You can find also this classification in some books. Polar, non-polar, it's exactly the same. When you say polar, it's hydrophilic or water soluble. When you say non-polar, it's lipophilic and it is water insoluble but plasma membrane soluble. And actually, the difference between polar and non-polar is their accessibility to cross the plasma membrane, as we all know. Examples of the non-polar or lipophilic اللي هم الستيرويدز هرمونز اللي هم مثل السكس هرمونز اللي هم estrogen, progesterone, and also the T4 اللي هم thyroxine, which also can access or can pass through the plasma membrane. So now let's finish here, and we will continue with our signaling lectures after about 2 weeks and a half. We will continue our subjects. Meanwhile, please try to identify all types of hormones, because this will make it easier to understand their mechanisms of actions. When you identify the chemical class, it is easier for you to identify their mechanisms of actions on cells. ويعطيكم العافية.